result of my PhD project. So ALS is characterized mainly by the degeneration of upper and lower motor neurons, but maybe you don't know that there is also non-motor symptoms, including autonomic dysfunction. So motor function is controlled by somatic system through the activation of striated muscles. And at the opposite, autonomic function controls involuntary function, like heart rate, blood pressure, or thermoregulation. The autonomic function is divided in two parts, with the parasympathetic nervous system, located in the brainstem and in the sacral part of the spinal cord, and the sympathetic nervous system, located in the thoracic and lumbar part of the spinal cord. The parasympathetic system is more involved in body basal condition, at the contrary to sympathetic nervous system that is activated in stress condition to fight or flight. These two systems have different outputs. The neurons, the parasympathetic neurons, connect the ganglia inside or near the organs. And at the opposite, the sympathetic neurons first connect postganglionic neurons in the paravertebral chains to control the target organs. So the output of the sympathetic system is the sympathetic preganglionic neurons, and they are organized in different nucleus, with the more important is in the intermediolateral area. And so now you have a better understanding about what is the autonomic nervous system. I will detail the non-motor system we reported during ALS. So it could occur at even before the first motor symptoms, and in some patients. We reported abnormal cardiovascular parameters like tachycardia, hypertension, decreased sweat and thermoregulation, disturbed gastrointestinal urinary function. And moreover, we know that autonomic nervous systems and somatic nervous system can't work themselves, and they are depending on each other. For example, when there is a muscular activation, the autonomic system is mobilized to offset the metabolic function. And moreover, we can consider that the sympathetic preganglionic neurons is the first cousin of the motor neurons, because they share some common points. They have the same embryonic origins. Their axon projects out of the central nervous system, and they are cholinergic. So the fact that there is a link between autonomic nervous system and somatic nervous system, and there is molecular similarity between SPN and motor neurons, we are wondering about the implication of the autonomic nervous system in ALS physiology. So our hypothesis is that the autonomic nervous system may be involved in the etiology of the disease, or that the degeneration of motor neurons eventually lead to the degeneration of autonomic nervous systems. To study this, the autonomic nervous system in this condition, we will focus first on the dynamic change in SPN during ALS, and then on the cardiovascular dysfunction in the SOD models. So first, to identify SPN in the IML area, we use a retrotracer through fluorogold injection. We did it on SOD1 models at two different ages. An early stage when there is the first motor symptoms, and a late stage where you can see some motor symptoms. So to select it precisely, the SPN in the IML area in the thoracic part of the spinal cord, we use laser microdissection, and we extract and sequencing the RNA of SPN in in each sample, and then we compare each sample with each of the other. So if we compare the SOD mice and the wild-type mice at the early stage, here we can see that there is already dysregulation, even if in the lumbar part of the spinal cord we have the motor neurons are not already degenerate. So we can see that there is some 100 genes downregulate and 100 upregulate, and these genes are encoding for immune systems. And then if we look at the mice at the late stage, there is an increase of upregulate genes with genes encoding for the complements, the cytokines, and the caspase. And so we can imagine that there is a strong reaction of the immune systems and the apoptosis in this condition. And we also saw that there is some ionic channels, including potassic channels, and some receptor and enzyme that are involved in sympathetic neurotransmission. So we can imagine that there is in this area some modification in the cell physiology. So our first results show that for the first time and at the very beginning of the first motor symptoms in the IML area, that SPN have a genomic alteration, and this genomic alteration is associated with an activation of immune systems and an alteration of the physiology of neurons and surrounding cells. And in the second part of my study, I work on cardiovascular parameter in the mice model, because we see some difference, some modification in ALS patients in cardiovascular parameter. To do this, we use telemetric approaches. We implanted a captor in the carotid arteria, the probe in the carotid arteria. This probe is a sensor of pressure, and it transduces the telemetric signal and it records the blood pressure, the heart rate, the body temperature, and the activity on a free-moving animal. We did some record on 4 different ages at the end of the disease to compare cardiovascular parameter. Because we know there is some modification of the cardiovascular parameter in mice according to the circadian cycle, we recorded the mice during 3 days to see the mice during their sleeping and their activity phase. And as you can see here, there is a difference between activity and sleeping phase. There is variation of the pressure. This is the record of 2 days, and you can see that there is a difference when the mice is sleeping or they are activate. At the end of this procedure, we did a special exercise. We forced the mice to swim during 5 minutes to see the effect of an intense motor exercise on the cardiovascular parameters. So if we look at the heart rate during, I will present only the first part of the recording, not the swimming part. And if you look at the heart rate during the record, you can see that there is effectively a lower heart rate in a light-on condition when the mouse is sleeping, and for the two genotypes, but we also see at the last recording, at the last stage, that we have a difference between the two genotypes, with a higher heart rate for SOD mice than wild-type mice. If we look at the blood pressure, we can again see a difference between the light-on and the light-off, with a lower blood pressure in light-on, and we don't see any difference according to the genotypes. And concerning the temperature, we have again a difference according to circadian cycle, and we also see that there is a huge decrease of the body temperature of SOD1 at the last stage, with 2 degrees less than the wild-type mice. So thanks to this method, we see that in the SOD1 mice model, we have an increase of the heart rate, like in human patients. And we see that there is a decrease of temperature, but we don't see the increase of blood pressure that there is reported in human. So there is still some work in progress. We will check. We will check whether the genomic alterations are associated with protein expression and function, thanks to Western blot and histology techniques. And we will check if the SPN are degenerated, thanks to immunostaining. And we will complete our observation concerning the cardiovascular modification by studying the sympathetic innervation of the earth with our collaborator Isabelle Brunet in Collège de France, and we will quantify and compare the blood metabolites after swimming exercise in SOD1 and wild-type model. So I would like to thank the ARSLA for funding, and my team and my collaborator, and you for your attention.
Thank you very much. Questions? Very interesting work. Have you correlated the blood pressure alterations with activity to know whether this is primary or secondary to an increased activity in sun mice, for instance?
We don't see, if I understand, I'm not sure that I could understand the question.
There are some papers, and actually we also have data showing that SOD mice have more activity in the inactive phase. So the question for me is whether the increased blood pressure, heart rate that you see could be secondary to an increased motor activity.
We don't see any difference between the activity of SOD1 mice and wild-type mice, but we have only 4 wild-type and 5 SOD1 mice, so now we can conclude. Okay.
Very nice, Maëlle. If I remember, the sympathetic axons are longer than the parasympathetic axons, right? They terminate closer to their end organ targets. And somebody from your city, Gwenelle Lemesson, has this very elegant work talking about mitochondrial energetics and the alpha motor neurons from ventral spinal cord. Do you know what the energy demands are for the sympathetic? You know, are they susceptible because they're as energy demanding as the alpha motor neurons, or it'll be different?
I don't know. I'm not sure to understand the question.
No. You have
Thank you for your talk. In humans, we observe in ALS patients sometimes also unexpected sudden death. So that's something that might be associated with autonomous failure. Do you observe something like that also in the SOD1 mouse?
No.
At the late stage, for instance?
No.
Maybe.
It's just to explain some in SOD1 mice. There is some interesting thing that when we're doing the swimming exercise at the last stage, the SOD1 mice is still okay to swim compared to some other mice that we have in our lab in the same exactly the same are dying before and have some much more motor symptoms. So I don't know if the exercise every 10 days is improve their life, but I don't have death before the end of the experiments.
Maybe I have a quick question, but maybe I missed something. So at the end, the heart rate is increased.
Yeah.
So do you think that the sympathetic tone is decreased, or the parasympathetic? I mean, homeostasis is a matter of balance.
Yeah.
So because at the beginning, your hypothesis was that the sympathetic system was impaired.
Yeah.
Isn't it? So at the end, your conclusion is that probably not, or that it's a matter of balance?
In one side, we study sympathetic neurons, but for the genomic part. But when we study the cardiovascular parameters, there is both. There is parasympathetic neurons and sympathetic neurons that we can conclude. We have to do more experiments.
And I have a quick one, because there is this, this very recent Norwegian study, more than 300,000 patients, and they showed that a low heart rate associated with high-intensity exercise was protective in men. I mean, again, the old legend of exercise as a risk factor in ALS. So do you have some insight about that? No.
I don't know.
No, I think that there is no response to this question. It was more.
Okay, thank you very much.
Thank you.